Thank you for coming. I don't speak Spanish, so I'll have to speak in English today. But it's my first time in Mexico City. Thank you again, Louis, Justine, for inviting me. I mean, it's such a majestic city, and I'm having a great time here. Thank you. So in my laboratory, I'm interested in understanding how cells respond to stress. And as you know, that we live in a harsh environment. And of course, our cells are exposed to so many bad things. And if we just leave it alone, then all our cells will decay. We're like these cellular machines. And if we were a car, then by now I wouldn't be functioning because I'm too old. I mean, but the thing is, we have this amazing ability, our cells have this amazing ability to detect and respond to injury and repair and stress. And if that machinery is robust, then we can live long, healthy. And if that machinery is not so robust, then we won't be able to live long and will decay after 5 or 10 years, just like a car or any other machines. And that's something that I'm interested in. And of course, there are many different stress response pathways, but the particular pathway that I have been working on for the past 15 years is a pathway called the unfolded protein response, or UPR. And what I try to depict over here is a cell. And a cell has a nucleus. And what I try to draw over here is a subcellular organelle called the endoplasmic reticulum. Now, the term unfolded protein response is a little bit of a misnomer because proteins exist in the cytoplasm, but also inside the endoplasmic reticulum. And this signaling pathway refers to a pathway that's activated specifically in response to protein misfolding in the endoplasmic reticulum. And as you know, proteins, they are initially synthesized as a chain of amino acids linked together, but in order to work as machines, they have to fold properly to form a shape of these particular machines that really make us live the life that we live. So folding is important, and if they fail to fold, then it's still okay, because there are all these signaling pathways that can tell the nucleus that there's something wrong in the endoplasmic reticulum. And inside the nucleus, we have our genome. And these signaling pathways should make the genome make more, transcribe more, and express more of the proteins that can solve this problem. And two particular pathways we've been particularly interested in. One is a pathway by these genes, and the other is a pathway by this PERK ATF4. And I'm going to today primarily talk about our more recent work on the PERK ATF4 pathway. And we use Drosophila for our studies. Now, in this field, lots of people are interested in the unfolded protein response, and most people use cultured cells, and they challenge them with chemicals that would cause damage. We have a slightly different approach. The other approach has been proven to be really productive, but by using an actual organism, we can actually address questions that other people have not been able to address. And for example, these pathways, they're also important for organismal development. And these pathways, they have disease relevance. There are diseases caused by excessive misfolded proteins in the endoplasmic reticulum that cause retinal degeneration, which we've been studying. And I'll tell you a little bit about our work on these pathways in lifespan and immune response. Okay. So again, key points that I want to get to today is how does this PERK ATF4 pathway work, the molecular mechanism, and how does this pathway affect things like lifespan and immunity, its biological effects. And near the very end, I'll tell you a little bit about the stress response pathways in development. I mean, we've been mostly thinking about these pathways in terms of stress imposed by environmental conditions, but apparently in our healthy tissues, we also have physiological stress, and we need these genes to deal with those types of stress. Okay. And why am I going to focus on this pathway mediated by PERK and ATF4? And the conceptual aspect that I want to get to is as follows. So most of us in our field were studying the stress signaling pathways that affect gene transcription. As you know, the DNA encodes the information, and in order for that to be realized, DNA has to be transcribed into RNA, and RNA has to be eventually translated into. But then there's some problems with it has to be eventually translated into. But then there's some problems with an over-focus on that, because we know that after transcription, now mRNA comes out to the cytoplasm, and it has to, this encodes an important stress response protein. So this has to be translated by these protein manufacturing factories called the ribosome. But then researchers also know that when there's stress, a lot of times this translation is inhibited. And I'll tell you more specific examples as we go along. But here lies the conceptual problem. These stress response pathways generated lots of important mRNAs, but they can't get translated. So why did this all happen, and how is this going to help the cell if they can't produce stress response proteins under conditions of stress? So to address this, to get a better insight, we decided to focus on a pathway mediated by PERK and ATF4 that does two things. It reduces mRNA translation, right? Tells the cell stop making new proteins, and it induces new mRNA. So by studying this pathway, maybe we can understand how does this work? I mean, they don't really make sense with each other. So before getting into the details, let me give you a very brief overview of our understanding of mRNA translation. So this, I try to depict an mRNA. This is the RNA that encodes information for proteins. And mRNAs have something that other RNAs don't have. For example, the M7G cap, we call this 5' cap. And this is important because this is where the ribosome gets loaded. This mRNA has to load the ribosome, other RNAs don't. And now the ribosome, when it comes, it shouldn't come empty-handed. It should come with an amino acid charged tRNA, methionine initiator methionine tRNA. And of course, there are other proteins that help load this really important bullet, if you will, to the ribosome, and that's the translational initiation factor. You know, the name doesn't really matter that much, but the name is EIF2. And it helps bring and load the initiator methionine tRNA to the ribosome, and it will load onto the mRNA at the 5' cap, and then now this mRNA can be translated and make proteins. Now, when there's lots of unfolded proteins in the endoplasmic reticulum, one of the ways that the cells try to protect itself is by telling the cell, well, there's too much unfolded proteins. The protein folding system needs a little bit of time to fold them. So in the meantime, stop making new proteins. Okay? And how does that happen? There's this ER stress sensor called PERK, and ER by endoplasmic reticulum. There are these misfolded proteins that get sensed by this protein called PERK, and when it senses it, then it phosphorylates EIF2 and inhibits its ability to bring initiator methionine tRNA to the ribosome, so then there's less protein synthesis. And the cells can now focus, instead of focusing on folding new proteins, they can try to fold these old proteins that couldn't really fold properly. But then something weird happens here. So most protein synthesis goes down, but there's this transcription factor called ATF4, and we're causing ER stress with a chemical called tunicamycin. And normally it's not translated, but only when other protein synthesis goes down, ATF4 protein synthesis goes up. This is a Western blot visualizing the protein. And how does this happen? And just briefly, this is I've tried to depict an ATF4 mRNA, ATF4 coding sequence over here, and the magic lies in the 5' UTR. There are these regulatory upstream open reading frames that normally block ATF4 synthesis when everyone is being synthesized happily. But then when everyone's synthesis goes down because this is phosphorylated, then ATF goes up, and that's because of these regulatory upstream open reading frames. I won't really labor too much, but ribosomes basically scan from the 5' cap, and when it sees this, it uses up the initiator methionine tRNA. And the empty ribosome continues to scan, and if it only gains the initiator methionine tRNA less efficiently, then it will bypass the second upstream open reading frame, and then it has a chance for the ATF4 coding sequence to be translated and recognized. That's why it occurs, and detailed mechanisms have actually been described by the work of David Rahn and Alan Hinnebusch, and much has been written about this topic by other researchers. So to sum up, I've told you about this particular branch of the unfolded protein response. When there's too much misfolded proteins in the ER, endoplasmic reticulum, then it activates this pathway, phosphorylates this translational initiation factor, so everyone's protein synthesis goes down. But then there's a weird transcription factor whose synthesis goes up under those conditions, and there's translational inhibition with new stress response gene transcription. And before David Rahn characterized this pathway, Alan Hinnebusch, the scientist at the NIH, discovered that this also happens when cells don't have enough amino acids. When they're starved, as a protective mechanism, they want to tell the cell, don't synthesize more proteins because we don't have enough amino acids. And it basically activates a different kinase called GCN2 that does the same thing, reduce overall protein synthesis, but then activate a transcription program by ATF4. Okay. So why did we get interested in this? We stumbled upon this while studying a different translational inhibitor called 4EBP. And let me explain to you how 4EBP works. This is the mRNA. And again, the ribosome has to load over here at the 5' cap. And the reason why ribosomes recognize the 5' cap is because there are other proteins that help it recognize it. The translational initiation factor called EIF4E. And then the ribosome gets recruited, it scans, and once it sees the AUG start codon of an open reading frame, then the other ribosome subunit comes, and then it starts synthesizing peptides according to this information. Now, 4EBP it stands for EIF4E binding protein, so it inhibits this process. So ribosomes can't really load to the 5' cap anymore, so protein synthesis goes down. And again, there are many stress pathways that tell the cells to reduce protein synthesis by activating 4EBP. And just here are a number of papers. There are a really huge number of papers indicating that various conditions actually activate 4EBP, but here's just a few that focus on the transcriptional induction of 4EBP by stress-responsive transcription factors such as FOXO. So, and now 4EBP is an EIF4E binding protein, and it inhibits cap-dependent translation. And there is a very specific biological phenotype that initially drew our interest, and which is, let me summarize these two particular papers. So these two papers were reporting that if you feed flies less yeast in their food, so they're getting less protein, then they live long. So this is a popular idea out there, let's not eat too much because, I mean, if we starve, then we're going to die all very early. But if we eat as much as we want, then we're also not going to live that long. That's a very now becoming an established idea out there. So let's be just a little bit slightly hungry all the time, then we'll be healthy. All right? And this, people have found that it applies to mice and flies and C. elegans and yeast. Just give them a little less than what they really want, and they live longer. And what these people have found is that when they feed flies a little less food, then they live long, and they live long because 4-EBP is transcribed. It goes up. But then they concluded that it's not really due to FOXO, it's due to some other signaling pathways that 4-EBP goes up. So we got interested in this accidentally. My old postdoc, Minji Kang, she was studying ER stress response, and one day she came up with a hypothesis. She noticed that FOXO is a popular transcription factor, and it's activated by Jun kinase. So one day she drew up a hypothesis. Well, ER stress must activate the stress signaling by Jun kinase, FOXO, 4-EBP, and I can get another paper. And she did this experiment in cultured cells, basically caused ER stress in these cultured cells by, again, treating them with ER stress-causing chemical tunicamycin. And she was happy. This is RT-PCR, basically looking at the mRNA levels, and she found that the mRNA of 4-EBP goes up, and this is quantification. But then she got really unhappy because when she got rid of FOXO through RNAi, it still went up. So FOXO had nothing to do with 4-EBP induction. Okay. And then she decided to ask, well, the starting point is ER stress, and there are nicely characterized ER stress responsive pathways mediated by PERK, ATF4, IRE1, XBP1. So let's ask if these unfolded protein response pathways are required. And she got rid of these mediators through RNAi, and what she found is that stress can no longer turn on 4-EBP transcription when these cells are lacking PERK and ATF4. Okay? So this led to the conclusion that ER stress activates PERK, ATF4, which now activates a second translational inhibitor called 4-EBP. Okay. Now, is that an interesting signaling pathway or not? And she decided to go back to the literature. Well, if flies eat less protein, then they can live long. And they can live long because 4-EBP goes up for some mysterious reasons, and the community doesn't know why it goes up. But we know that amino acid deprivation activates GCN2, that reduces translation, activates ATF4, and we just found that ATF4 induces 4-EBP. So maybe this is the new hypothesis. And she decided to test this, and she obtained ATF4 mutants that have been around since the 1940s. And she did this experiment. She took these mutants, and when she starves them, 4-EBP goes up. Okay? This is what other people have reported. When she gets rid of ATF4, 4-EBP doesn't go up anymore, which supports this hypothesis that starvation induces 4-EBP through ATF4. And this is a quantified data based on RT-PCR, basically showing the same thing. What about GCN2? And she, is this hypothesis still really true? She tested GCN2. This is a tissue, intestine, and when she starves them and she measures 4-EBP mRNA, now 4-EBP goes up. Other people had reported this. But when she gets rid of GCN2 through RNAi or ATF4 through RNAi, 4-EBP doesn't go up anymore. So this data supports this idea that starvation induces 4-EBP through this pathway. And then she generated GCN2 mutants. And these mutants, flies actually survive, and initially they look okay without the GCN2 gene. And she noticed that previous studies that looked at 4EBP mutants, and the phenotype is they look okay, but they don't live longer just because they ate a little less. That's the 4EBP phenotype. So she decided to test whether GCN2 has a similar phenotype or not. And she generated this mutant, basically deleted this region. And basically she found that GCN2 mutant phenotype is similar to the reported 4EBP mutant phenotype in that the wild type flies, these are the flies that have been on a high protein diet. Their survival curve goes like this. And when they're given a leaner diet, less yeast diet, less protein diet, then these flies live longer. But in GCN2 mutants, it doesn't matter whether flies eat really high protein diet or low protein diet. There's no benefit of eating a little less, which indicates that GCN2's role is to help, it contributes to the flies living longer when they're eating slightly less rich diet in their food. Okay. So the pathway, the link that we've uncovered appears to have some connections with lifespan, and it has to do with detecting nutrients in our diet and dictating how long or how short we live. Now, we have an ongoing story that's currently under review in Cell Reports, and maybe I can tell you a little bit about that story. It's basically the same pathway, but it also is involved in innate immune response. Okay, so the starting point is as follows. So lots of different types of stress reduce protein synthesis in our body. I mean, perhaps the best known example is viral infection. It shuts down protein synthesis for our cellular mRNAs, but viral RNAs can still be translated. So they turn our cells into a virus particle manufacturing factory. That's how, and that's very well established. But researchers have also found that bacteria, not virus, but bacteria also reduce our protein synthesis. And just to show you a few examples, this paper published in 2012 showed that in flies, if they feed flies pathogenic bacteria, then GCN2, the gene that we're interested in, is activated and protein synthesis goes down. Now, even before that, a different group had found that if they infect flies with pathogenic bacteria, then 4EBP goes up, is more synthesized. And no one had any idea that these two might be connected. But now we know that 4EBP is an ATF4 transcription target. People have known that ATF4 lies downstream, so we decided to test whether things work through this signaling pathway. And this is the work primarily of Deepika Vasudevan in my laboratory. And she utilized the reporter that Minji had made. So we were, by that time, pretty sure that 4EBP is a direct transcriptional target of ATF4. So this is a transcription factor. So it induces target genes by binding to DNA regulatory sequences. And this region of the genome has ATF4 binding sites, as shown here. And Minji had made an artificial reporter, basically cut out this region and put it upstream of the red fluorescent protein called DS-RED. And now this DS-RED gets induced when cells are starved of amino acids. And we know that this is due to ATF4 because if we knock down ATF4 through RNAi, then the reporter doesn't get activated anymore. And we can make a fly out of this, and it's detecting signals even during development. Now, this is, I don't know if you can see because there's no ATF4 signal here, so no fluorescence. And then gradually it comes on, and it correlates with the fly larvae stop eating. So when they're growing, they're eating all the time. There's plenty of amino acids, so there's no signal. When they're about to become pupa, then they stop eating. And of course, there's amino acid deprivation, and you see lots of signals here. And we're pretty sure that this is all due to ATF4 because we take this, which is here, and we knock down ATF4, then the red signal disappears. So this all red signal is due to ATF4 activity in normally developing flies. And Deepika was now able to take those flies. Now this is the adult intestine, and she either fed them just normal food or food with pathogenic bacteria. And the pathogenic bacteria that we're using is something called Erwinia carottova 15. And what she sees is that this ATF4 4EBP reporter lights up in the gut and in the fat body. This is another tissue, indicating that pathogenic bacteria has something to do with activation of this pathway. And she did, just to double confirm the results, she did RT-PCR against 4EBP. In normal flies, now 4EBP transcript levels go up in wild type or in other mutant backgrounds, but in ATF4 mutant backgrounds, they don't go up, supporting this idea that ATF4 mediates 4EBP induction in response to pathogenic bacterial infection. Now, what we do often is when we see something, we have to ask, well, does it matter? Does it really help flies fight infection? And to do that, we did this pathogenic load experiment, which is basically feeding flies with pathogenic bacteria and allow them half a day, and then see how much pathogenic bacteria we can recover from the fly's body. And normally, I mean, if one is controlled for a wild-type fly, then 4EBP mutants, they're not able to clear pathogenic bacteria that well, so we detect a lot more pathogenic bacteria in their body. ATF4 also, we recover a lot more pathogenic bacteria, indicating that these genes are required for innate immunity of the flies. It's not only just turned on, but it's functionally required for innate immunity. Now, that was all good, but then initially we couldn't make any sense of it because what do we know about innate immunity in Drosophila? Now, this is primarily the work of the Nobel Prize winning group, Jules Hoffman and others, his colleagues Bruno Lemaitre and many others in the field. They figured out this pathway of innate immune response pathway. Basically, there are these proteins that detect bacteria, activate transcription factors, and very easy, these transcription factors induce antimicrobial peptides, and they're going to be synthesized and they're going to kill bacteria, and that's the end of the story. Now, our result was puzzling because 4EBP is a translational inhibitor. So and 4EBP is helping with the immune response. Now, if 4EBP is inhibiting antimicrobial peptide synthesis, then it would actually make flies worse when they're dealing with pathogenic bacteria, but we found the opposite. This translational inhibitor is helping with the innate immune response. And to really understand the relationship between 4EBP and these well-known antimicrobial peptides, Deepika did RT-PCR experiments, and she found that 4EBP mutants in gray, wild type, there's not much of a difference between the transcripts of antimicrobial peptides. So what is it? I mean, the field tells us that it's the mRNA induction that plays an important role in antimicrobial peptides. And Deepika decided to look at the protein levels. So she basically infected these larvae, collected their larval blood, hemolymph, subjected them to mass spec analysis, and what she found was that these are all these different antimicrobial peptides. They're found at much higher levels in wild type, but when there's no 4EBP, they're a lot lower. And again, this is the opposite of what you would have expected from a translational inhibitor. In the absence of translational inhibitor, we would have expected more protein synthesis, but we're actually seeing less antimicrobial peptide synthesis. And probably this is why they can't fight pathogenic bacterial infection that well anymore. So how can that be? And so let's review. So we had an idea, we had a hypothesis, and let's go back to the normal function of 4-EBP. 4-EBP is a protein that inhibits EIF4E and loading of ribosomes to the 5' cap. And most people think that most cellular mRNA translation requires EIF4E and loading of ribosomes to the 5' cap. But in case of, for example, viral RNAs, they have a different way of recruiting the ribosomes. They don't care, viral RNAs don't care about any of this because they have this sequence called iris that would independently bring ribosomes and continue to synthesize, translate their RNA, even if the cellular cap-dependent mRNA translation is shut down. So we started hypothesizing perhaps there's something similar going on with antimicrobial peptide mRNA. So is it possible that antimicrobial peptides have also 5' UTRs that contain iris? And we set up a collaboration with Michael Marr, and they have an assay. They make this mRNA. It has 2 cistrons, 2 reporters, and the ribosome would load here at the 5' cap, and it usually synthesizes the first open reading frame and it falls off. But then if there's a iris sequence over here, then that would independently recruit another ribosome and firefly luciferase gets synthesized. And now this is the assay to see whether our favorite DNA has iris or not. And in our first collaboration, he reported that ER stress responsive transcripts, like this is the ER chaperone that's expressed when cells are stressed with lots of misfolded peptides in the ER, it scores positive in this iris assay. And we extended this analysis to antimicrobial peptide 5' UTR. This is, again, our version. Dipika subcloned, introduced antimicrobial peptide 5' UTR between GFP and DS-RED. And what she finds is that if there's no iris, or if she puts the 5' UTR of Drosophila in the antimicrobial peptide in an opposite orientation, DS-RED never gets made. But if this is placed in the forward orientation, then DS-RED is synthesized, suggesting that this antimicrobial peptide's 5' UTR has internal ribosome entry sites, iris elements, just like the virus. So how do we explain all this? Well, just to summarize what I've told you so far, I told you we started out from our interest in lifespan. 4-EBP is induced when flies are fed a little less, and they live long because 4-EBP is induced. And the pathway was not known, but we think it's the GCN2 EIF2 alpha 4 ATF4 pathway that mediates 4-EBP induction. We also found that pathogenic bacteria also does the same thing, and it helps flies resist that kind of a different stress, dealing with pathogenic bacteria. And how can 4-EBP really help cells fight stress? Well, most mRNAs are synthesized through cap-dependent translation. EIF4E helps the ribosomes load at the 5' cap. And 4-EBP, when it's induced, it inhibits this. And now there are more resources left, more ribosomes available. And those will be now diverted to internal ribosome entry site containing mRNAs, such as stress-responsive transcripts or antimicrobial peptides, and therefore their translation paradoxically goes up, and that enhances stress resistance in the fly. That is our working model. Okay, so in the remaining 15 minutes, I want to tell you about physiological stress. And this is another story that's now in press in Journal of Cell Science. We were very interested because most people study stress response in the context of exogenous stress. For example, chemicals or mutant proteins. But it turns out that our healthy tissues also have a little bit of physiological stress, and it requires the help of these stress response pathways. Our latest work I think, really pounds that point. So this is related to everything that I told you earlier. So we've been focusing on PERK-ATF4 pathway. And now we're going to talk about the other pathway, but they're related. Because if this pathway is not working, things are going to get worse here, and it's going to activate this other pathway. All right. So this IRE1 XBP1 pathway, how does that work? There are misfolded proteins in the ER, and they activate a different sensor called IRE1, and it has an RNase. So it cuts, what it does is it cuts this XBP1 mRNA, and it undergoes a splicing reaction. And this spliced XBP1 now encodes an active transcription factor that goes to the nucleus and induces chaperones and other proteins that would help get rid of these misfolded proteins in cells. And reports that have been emerging in the past 6-7 years indicated that IRE1 has other RNA substrates, and they're primarily degraded. They don't undergo splicing, but degraded. And there are also reports that IRE1 does other things than just using its RNAs. And these are based in cells, and whether this is going to be relevant in our body, in an animal body, is unclear. So years ago, we decided, well, let's use flies to study this. And what is known as ER stress activates IRE1, and it induces XBP1 splicing. So we can make a reporter for this. We can, after splicing, there's a frame shift in this translation. So we can put GFP in a certain reading frame, and normally GFP is going to be translated out of frame, and we won't see any green fluorescence protein. But if there's stress and IRE1 splices this, the reading frame changes and GFP gets expressed. And this reporter turned out to work very well. If we challenge the cells with ER stress causing chemical, then this GFP appeared. And if we get rid of IRE1 through RNAi, this reporter did not appear. And using this, we showed that this pathway is relevant to disease. So there's a human disease called autosomal dominant retinitis pigmentosa. People who have this disease, they become blind when they're 40, 50 years old. And that's because they've inherited a disease mutant allele of this gene called rhodopsin. And flies suffer from the same disease. Flies also have rhodopsin, and they have these mutations. And if the flies inherit this, then they go blind when they become old, which is 30 to 40 days. So we can actually study this disease in the fly. The absolute time doesn't matter. It's really about whether it be a fly or a human, whether they're old or young. And we basically we had this ER stress reporter, so we had decided to test whether this disease has anything to do with rhodopsin misfolding in the endoplasmic reticulum. And we put our reporter in the background, and this is now the fly eye cross-sectioned. And what we see is in the disease model, the endoplasmic reticulum stress reporter is active. And in healthy flies without the disease mutation, there's no ER stress reporter activated. And okay, there's ER stress, but does it matter? Does it affect the course of retinal degeneration? And we found that yes, it does, because if we get rid of one copy of XBP1, then the flies degenerate their photoreceptors much faster. So normally, the fly photoreceptors with the disease mutant, they're okay until 16, 20 days, but without one copy of XBP1, now even at 6, 12 days, there's significant degeneration. So that was our prior work. Now, physiological stress. I personally became interested because I always understood these pathways as stress response pathways. There has to be stress coming from the outside. But these mutants, they die only after first instar larval stage. We didn't stress this at all. We tried to take care of them really well, fed them well, and they all died at this stage. So, and initially we couldn't believe this result, so we put back this XBP1 DNA, IRE1 DNA, this genomic DNA, into these, and now they can survive to adulthood. So they're dying because they don't have IRE1. Okay, so where do they require IRE1? And we had a simple strategy. So sorry. If there's a cell, a healthy cell that requires IRE1 because there's low levels of stress in the ER, and if we get rid of it, then things are going to get worse in here. And because things are getting worse, it's going to fire other pathways even strongly, PERK-ATF4 pathway. Now, we have a PERK-ATF4 reporter, 4-EBP intron DS-RET, that I talked about earlier. And we made a fly. We got rid of IRE1, and this is now, I don't know if you can see, but there's a larva here, and it has IRE1. And there's another larva here, which doesn't have IRE1. And this reporter is now blazing red in the larva that doesn't have IRE1. Okay? So this means that, yes, they're dying at this stage because they require IRE1. Otherwise, there are lots of cells that are very unhappy. And we generated now IRE1. The way it works is it detects misfolded peptides through its luminal domain. And once it detects it, now it has RNase activity that does a lot of things. So we generated now mutant versions that can't detect misfolded peptides, mutants that cannot cleave RNase. And what we find is that only wild-type IRE1 can get rid of stress. All these mutants that can't detect misfolded peptides or can't detect RNase, they can't get rid of stress. So this indicates that during normal development, there must be misfolded proteins that has to be dealt with, and then activate IRE1's RNase activity to deal with that stress. And XBP1, so does it really require the downstream gene XBP1? And again, in XBP1 mutants, this reporter lights up, right? And what we did was XBP1 gets cleaved by IRE1 in this target sequence. So we rescued this fly by putting back a wild-type XBP1, and now there's no sign of stress. But when we introduced this silent mutation, so it no longer can be spliced by IRE1, but the amino acid sequence is the same. These are called silent mutations. Then they can't help these flies. They all die. And there's strong signs of stress, indicating that during normal development in these tissues, there are tissues that require IRE1 mediated splicing of XBP1 to deal with normal physiological levels of stress. And dissection of these tissues basically show that it's not all tissues, but really specific parts of the fly anatomy that requires XBP1. In the absence of XBP1, only these red tissues light up, meaning that these are the specific tissues that are suffering from physiological stress, very low levels of physiological stress, but they're there, and we're able to visualize them with our reporters. So to sum up this part, I told you that this pathway, IRE1, detecting misfolded peptides and doing a lot of things, it's also required for normal development. And IRE1's ability to splice XBP1 is absolutely essential for development. And IRE1's RNase function, what we know is that RNase function is absolutely required for normal development. And which means we haven't disproven the role of RNase independent functions, but it's either masked by the RNase dependent function, or at least we found no evidence to support RNase independent functions of IRE1 in the context of normal development. So with that, I'd like to thank the people who contributed to the work. I tried to acknowledge them as I went along. Minji Kang, a former postdoc, she now has her own lab in University of Ulsan in Korea, doing well. Deepika Vasudevan is the one who has really pioneered the work on innate immune story, and she's right here. And I didn't really have a chance to talk about it, but Huaiwei Huang over here, he has a different story about the role of retinoic acid signaling in unfolded protein response, and other people are working on other less complete stories that are in the pipeline. Some of the collaborators, David Ron, Pedro Domingos, Michael Marr, Heinrich Jasper, and also the funding. Thank you for your attention, and I'd be happy to try to address any questions that you might have today.
Bueno, no sé si alguien quisiera comenzar con las preguntas.
Well,
Yes.
thank you. Thank you for your presentation. Congratulations. I have a question. In your opinion, what is the most important physical-chemical parameters in the control system? Time, concentration, pH, temperature, and why?
Right. So I don't know if I can pinpoint one, because in my opinion, they're all important, right? So these stress response pathways, definitely there are time course of events. Every signaling pathway that I told you about, these in general, in cell biology, these signaling pathways, they all have activators and then feedback inhibitors. Almost all pathways have that. So time course is very important, because in the first 12, 24 hours, cells are able to deal with it really well, the stress. But chronic stress, they're usually not so good because of these negative feedback inhibitors. Temperature, I would say in mammals, it's less of a problem because we're all our cells are all bathing in 37 degrees. But the unfolded protein response, when we analyze these pathways in the fly, temperature does matter. Sometimes the phenotype is stronger in 25 degrees and less strong in 20 degrees. It definitely matters, but in mammals, in humans, I'm not sure whether temperature will matter as much as cold-blooded animals that live out in the wild. And you know that all biochemists will tell you that pH, yes, it's important, because you have the wrong pH, proteins are not going to fold that well, and of course then they'll fire up the unfolded protein response, heat shock response. Concentration is probably also important. I think Justine and Louis will be able to tell you that you vary the concentration of any toxic compounds, then there's always, a low threshold concentration that cells can cope with. But then toxic compounds, after a certain concentration, even if we have robust stress response signaling, we won't be able to deal with those. I think they're all probably important in my opinion.
¿Otra pregunta? ¿No?
Thank you. I was wondering about the other pathways sensing ER stress.
Mm-hmm.
Are they also essential for development in Drosophila? Is this known? And if
Yes.
so, the phenotype is similar to the one of the
Right.
the
So-
flies that are deficient for RI?
Right, so
A?
what we know is PERK, ATF4, they're all essential for development. Now, we haven't, that's our next project, but we haven't characterized in as much detail as the loss of IRE1. Now, it's not just flies, because PERK mutation is actually associated with a human disease, and it's a developmental disease. It's called Walcott-Rallison syndrome. So once in a while, there are these people who lack the PERK gene, And these infants are born and they suffer from diabetes, and they have problems in the pancreas and the skeletal bones. And part of the reason is because if you think about it, the pancreas, pancreas acinar cells, they're really busy secreting, synthesizing tons of secretory enzymes and secreting them through the endoplasmic reticulum and Golgi, the secretory pathway. Beta islet cells, that they're really busy. They have a very high load of synthesizing insulin and then channeling them through the secretory pathway. So looking back, of course, those are the cells that are likely to suffer from physiological stress. And of course, without PERK, those cells are likely to encounter problems. And Walcott-Rallison syndrome, one of the phenotype is these beta islet cells in infants, they start dying out and they start developing type 1 diabetes. And there are similar studies of PERK knockout mice that also report problems in the pancreas. So they're essential, and we'd like to delve more into it using fruit flies.
And what about aging? Is there a relation between the UPR and the lifespan? Can this be tested in flies?
The GCN2 is not exactly UPR, but it's amino acid deprivation response, and I showed you that does have an effect on lifespan and aging. Now, the literature, there are papers out there arguing that various branches of the unfolded protein response do affect lifespan in model organisms. And for example, there's a guy called Andrew Dillon. He's a big figure in proteostasis, protein homeostasis and aging. He primarily uses C. elegans for his research, and 3-4 years ago, he had a Cell paper showing that if they hyperactivate XBP1 in the worm, then they live long. And I think people have not so difficult time accepting those ideas, because I think a lot of people believe that, so what is aging? Well, we deal with all these different bad proteins and bad environmental factors, but when our cells are young, robust unfolded protein response can deal with it. They can get rid of these bad proteins really effectively. And the bad proteins, they're there at similar levels, but what changes is our cell's ability to deal with those bad proteins. And of course, if we jack up unfolded protein response in old cells, then they're going to behave more like young cells. That's a very popular idea that's out there currently.
Thank you.
¿Otra pregunta? Pueden preguntar en español, si gustan. No? I have 2 questions, actually. It's a general question. I'm going to switch to Spanish because of that.
All right.
La primera pregunta es, se conoce, por ejemplo, los priones. Los priones que provocan la enfermedad de la vaca loca. ¿Alguna de las vías que nos mencionó están activas durante este tipo de enfermedad, por ejemplo?
Okay, it's buzzing a little too much, and I can't really.
Okay, about the prions.
Yeah.
Do you know if
About the what? I'm sorry.
the prions?
Prions.
Sorry. You know if it exists, these 2 pathways are turned on in that case?
Mhm.
Or not?
Yes, I think some prions, there are literature indicating that prions impose stress in the endoplasmic reticulum and activate the unfolded protein response. And there are people who study that connection as well. We haven't looked into it ourselves, but I wouldn't be too surprised, because they eventually cause misfolded aggregates. And there are even reports that unfolded protein response can be indirectly activated by aggregates even in the cytoplasm. And we have had one positive result with alpha-synuclein. I mean, it's a cytoplasmic protein, and for some reason it activates the unfolded protein response that's supposed to specifically respond to ER stress. So I wouldn't be too surprised if prion also activates this pathway.
Okay. And the second one, it's conoce. I'm gonna translate it. ¿Conoce algún compuesto o un contaminante que pueda formar agregados proteínicos en el organismo, en un organismo?
It was buzzing too much, sorry.
Okay. Do you know any kind of disease, well, I mean, contaminant that could set up this kind of response also?
Contaminant, meaning chemicals.
Chemicals or
antibiotics. Right. So, well, the favorite, I mean, one of the most popular chemical that people use to activate the unfolded protein response is tunicamycin. And tunicamycin, I guess it started out as an antibiotic, but at a high dose, I mean, it's a really toxic compound. What it does is it interferes with the glycosylation of proteins. And glycosylation is something that happens in the endoplasmic reticulum. And lots of proteins require, N-glycan residues in order to fold properly, because the chaperones, they recognize not only the misfolded peptides, but they also recognize the N-glycan groups. So that's one example of a very popular chemical that would cause stress and make unfolded proteins in the endoplasmic reticulum. I'm pretty sure that there are many chemicals that would impose stress in the endoplasmic reticulum. For example, I wouldn't be surprised if mercury would do the same thing, because mercury, at the end, it modifies proteins as well. And if protein folding is interrupted, then we would expect unfolded protein response to be activated.
We'll see.
Yeah, we'll see. Yeah.
Gracias. Pues sí.
I can say it in English,
Okay.
in English first. When you're talking about physiological stress, what are you talking about specifically, and why we can't relate that to like an environmental stimuli?
Why we can't?
Or we can? Or?
I guess they could be affected by environmental stimuli. I mean, even simple things like, I'm sure, the amount of diet. I mean, if we have high protein diet, low protein diet, that's going to affect the same pathway, right? Because PERK and GCN2, they do the same thing. GCN2 responds to low protein diet. PERK responds to the unfolded protein response, and they activate the same pathway. So I wouldn't be surprised if environmental factors are going to affect the degree of physiological stress. I mean, for example, PERK mutations and Walcott-Rallison syndrome disease. I'm pretty sure that, I haven't really looked into that disease that carefully, but I'm pretty sure different babies have different onset in the time of onset of the disease. And some of them are likely to be affected by environmental reasons. That's my guess. But I guess it's currently, I feel like it would be a reasonable guess, but I don't think we've really demonstrated that point in one way or the other.
Pues si no hay más preguntas, muchísimas gracias por la presentación y gracias a todos también por haber asistido.